Great, and for our ultimate talk of the afternoon, Dr. Loren Pena is an assistant professor in the Division of Medical Genetics at Duke University in Durham, North Carolina. She's going to give a presentation on new genetic discovery and other insights from diagnostic evaluations in the Undiagnosed Diseases Network. Dr. Pena, thanks.
So thank you for inviting me to talk. I'll be presenting some of our work at Duke as part of the Undiagnosed Diseases Network, or UDN. We've done some exciting work, and I think it's all thanks to the gift of protected time and funding. And so this is partly an infomercial because the protocol is open for enrollment, but I wanted to present what we've done so far. And I have no conflicts of interest to present. So I think I'm preaching to the choir here when I say that rare diseases are a big challenge, and collectively they affect a large number of American citizens. And of course, we all know that they present a pretty big challenge in terms of recognition, diagnosis, determination whether this is a presentation of a common condition or a symptom of a common condition or part of a syndromic presentation. And there's a number of medical and socioeconomic and psychosocial burdens that are presented, partly because many times these diagnoses become what we frequently call a diagnostic odyssey. And so characterizing and diagnosing undiagnosed conditions in some ways is the law of diminishing returns. And so about 50% of the patients that we see can benefit from an evaluation and targeted testing. And some work from our group has shown that usually diagnosis, or the majority of diagnoses, occur at the first visit. And then when the patients don't reach a diagnosis and start to come back to clinic, the frequency of diagnoses just becomes lower and lower. And so this is where the UDN comes in, because the protocol really provides a mechanism for comprehensively evaluating these patients that are the ones that are past the initial or the second evaluation, and really the prospect of a diagnosis becomes more and more difficult. And so the UDN is an offshoot of the Undiagnosed Diseases program that started at the NIH in 2008. And so we started accepting patients in 2015. And so Duke is one of 7 clinical sites that are listed in red. We operate in partnership with Columbia University. The PI at Duke is Dr. Vandana Shashi, and the co-PI is Dr. David Goldstein at Columbia. In addition to the clinical sites, we have a biorepository, 2 sequencing facilities that offer whole exome and whole genome sequencing. There's a metabolomics core facility. There's a model organism center that can provide model organisms work in flies and in zebrafish. And then coordinating all this is the coordinating center at Harvard. And so far, since just under 2 years, the program as a whole has evaluated almost 1,600 patients. The admission rate is about 40% across the network. We accept patients across the age spectrum, so pediatric and adults. I will say, though, that the majority of the adult applications that we receive lack objective findings, and so the acceptance rate for adults is much lower. And so far, in the 203 applications that we have received and 82 that we have accepted, the diagnosis rate at Duke is about 37%. So it's a little bit higher than the network as a whole. And I'll present some of the work that we've done that really has enabled us to reach a higher diagnostic rate. And so I'm going to present 3 cases that we've seen. And the first one was a patient that had whole exome sequencing when she came to us. And it was really going back to deep phenotyping to narrow down what we thought the differential diagnosis was, and then to develop a diagnostic algorithm for that patient based on the phenotype. And so this is a 3-year-old Pakistani girl who had developmental regression that started at about 16 months of age, and the parents were consanguineous. When we saw her, she had lost all motor milestones, she had lost the ability to vocalize, and she couldn't feed herself. There was a family history in another consanguineous marriage in the family of 2 children who died at 2 or 3 years of age with neurological deterioration, but we didn't have any more information besides that. And so, as I mentioned, she had a trio exome that had previously been done, and that had shown a homozygote variant in the RP-GRIP1L gene that's associated with a minority of cases of Joubert syndrome. And she'd also had mitochondrial genome sequencing. She had an array as well, a SNP array that proved to be very, very helpful, and I'll talk about that in a minute. As part of the evaluation, she had an eye exam that showed bilateral optic atrophy. She had a neurological exam, and she had been seen by Duke Neurology previously, and they noted that her neuro exam had changed over time. And so even though initially she'd had hyperreflexia and spasticity, now she was profoundly hypotonic, she had tongue fasciculations, she had reduced tendon reflexes. She'd had an MRI in the past that showed cerebellar atrophy, but other than that, there was no molar tooth sign, and really the phenotypic fit for Joubert based on the brain MRI and the physical findings was poor. So we didn't think that the variants in RP-GRIP1L were the answer. And so we did a reanalysis utilizing the raw data from the exome, and that was done through our bioinformatics partnership with Columbia. And there were no additional variants of interest. We went back to the SNP array, and since the parents were first cousins, we looked at the regions of homozygosity and really looked at any autosomal recessive conditions in those regions of homozygosity that could fit the phenotype. And we came up with 5 conditions, and 2 in particular we were very keenly interested in. So PLA2G6, which causes infantile neuroaxonal dystrophy, and ACO2, which causes optic nerve atrophy and cerebellar atrophy as well, although the onset is a little bit earlier than for this child. And so we decided to actually have those 2 genes, PLA2G6 and ACO2, sequenced by a different method, so not next generation sequencing. And luckily, the University of Chicago offered Sanger sequencing. And so what they saw is when they tried to amplify exon 1 and PLA2G6, there was actually no amplification. And they were able to map the breakpoints and actually confirm that it was a deletion that was about 2.4 kbs that included the promoter and exon 1 of PLA2G6. And this is a plug for academic labs, because they were an academic lab. They were able to do a little bit more lab of the what I call custom work. And so they did RT-PCR to confirmed mRNA expression from that gene, thinking that there should be no expression of the promoter and exon 1 were both deleted. And so in fact, there was no mRNA expression from PLA2G6 when compared to control in this child. And so the diagnosis of PLA2G6 was confirmed for her. The phenotypic fit was good. There was a mechanism for this. But if you recall, she had whole exome sequencing and reanalysis of PLA2G6. So we went back to the lab and asked why this hadn't been picked up in the exome. And so it turned out that not only is exon 1 coverage not traditionally the best by next generation sequencing, but actually because often it's not a coding exon, many captured kits will actually exclude it from the kit, so it wasn't covered at all. So this was a lesson to us whenever we have genes of interest and we look at overall coverage of the specific gene, and we think that 97%, 98% is good, that we really have to ask very specific questions about coverage of each exon. And of course, that the good old phenotype is really where the answer was, and for this child it was just very helpful to utilize the information that we already had to look at her symptoms. And so the next 2 cases that I'll present are cases that had negative exomes but actually had candidate genes. And so the question here is, how do we prove that this one candidate gene is actually causing symptoms when we only have 1 patient? And so the first patient is a 20-month-old who had postnatal microcephaly and cataracts, and he had infantile spasms that were very difficult to control. He had profound global developmental delay and also delayed myelination on a brain MRI. And he had an exome in trio that was not diagnostic. And so, again, in partnership with Columbia, we had the exome reanalyzed and looking at specifically the bioinformatics signature of any candidate genes that would come up, as variants of interest. And so, in that list, there were 5 genes that came up, and 4 of them had not been previously described in human disease or in constitutional mutations. And so one in particular, NACC1, was of keen interest to us. It was a de novo change for this child. This protein has been described as a transcriptional regulator. It had been previously reported as being overexpressed in multiple types of neoplasms. And so our bioinformatician Nick Stong was able to look at the bioinformatics signature for the NACC1 variant. And so, of course, the first thing that he did was ask whether the change had been previously described in any of the control samples, and it hadn't been present in any of the internal or external control databases. There was strong conservation of the amino acid at this specific site. And just to give you an idea, the highest GURF score, the highest conservation is a 6, and so he was pretty high it was really pretty high at 4.9. The change was quite deleterious, and it was predicted to be one of the, less than 1% most damaging variants in the genome. There were several in silico prediction algorithms that were used, so mutation taster, SIFT, polyphen, and all of them actually gave them a score that was damaging or probably damaging. The gene product was expressed in neuronal tissue, and the gene is quite intolerant to loss of function mutations. So it was looking like a pretty compelling candidate, but I think in a court of law, a lawyer would say that this is circumstantial evidence, and so we really needed to, get beyond one case. And so one of the things that is available to us as part of the UDN are the patient pages, and so this is with consent of the families, and so whenever there's a candidate gene, the UDN will buy the internet domain, and so whenever somebody's searching for the gene name, this patient website will come up. And so there is a photo of the affected child, there is a description of the child and the symptoms, and there is a contact us site button where people can actually reach us. And so with the assistance of the web page, the core sequencing labs doing their own searches in their sample databases to see if they have any other patients. And also by listing the change in gene matcher, we were able to come up with, I think it was 7 patients in total. And so when we started to compare notes of all the patients that we had as a result of, essentially social media, the first observation was that everybody had the same exact mutation in the gene. Everybody was de novo, everybody was constitutional except for one patient that had mosaicism in the blood. They all had profound developmental delay. They all had seizures of different types that were very difficult to control. The younger ones had evidence of delayed myelination. The older ones had evidence of volume loss. And so this looked like a pretty compelling story to us. And some of the other, some of the additional information that we used was, calculating a p-value to see if, what the likelihood was of finding this change in a cohort of 7 children with the same mutation and the same gene. And so I think that the calculation came up to be about 10 to the -6. So it looked like, there's a very low chance that these findings in common in a cohort of 7 patients was pretty low. And so we published this as a new disease gene. in patients with delays and delayed myelination, cerebral atrophy, and difficult to control seizures. And so the model organisms work is really a crucial piece of information, and that is ongoing, but understandably it takes a lot longer. And so it turned out that one of the family members of one of these children is actually a neuroscientist. And so that group is actually undertaking the mouse work, and to this date we don't know what the function of this gene is. And so the second case that we had that had an exome that was negative was an 8.5-year-old boy who had macrosomia, and he had some dysmorphic features. He also had some minor congenital heart disease. He had multiple capillary hemangiomas, including a glabellar hemangioma in his forehead. He had some developmental delay, and he had some autistic features. He'd had extensive evaluations that I've listed here, and an exome that was done in 2012 that was not diagnostic. And so because he had an exome that was done in 2012, we decided to repeat the exome as a trio, to really look very closely at any candidate genes that could come up. And so there was one change in particular in the ASXL2 gene. It was a frame shift mutation that was of interest to us, and actually that variant had been on our watch list for this child based on the exome results that he had in 2012. And so there had been no role in disease in humans that had been assigned to ASXL2 previously, even though it has been reported as being overexpressed in several different types of tumors. There are an additional 2 members in this gene family, ASXL1 and 3, that are known to cause neurodevelopmental syndromes. So ASXL1 causes Boring-Opitz syndrome, and then ASXL3 causes Bainbridge-Roper syndrome. But these children tend to be small, and our patient was macrosomic. The mechanism for disease in ASXL1 is thought to be haploinsufficiency, and for ASXL3 it's a little bit debated right now. There's some publications that posit haploinsufficiency versus others that say that it could be a dominant negative. And so we essentially followed the same path that we did for the previous case, and so we listed it on GeneMatcher. We created a patient page for our patient, and we had our core labs actually take a look and see if they had any other patients who had changes in ASXL2 reported as a gene of interest. And so pretty quickly, I think within a matter of a month to 6 weeks, we had 6 cases. And when we started to compare pictures and compare notes, some of the characteristics became a common theme. So everybody was either macrosomic or had macrocephaly. They had prominent eyes. They all had this glabellar hemangioma. There was some congenital heart disease as well. And so, we felt like this was a compelling story. Again, our bioinformatician did a chance calculation for the possibility that we could have detected these variants de novo in ASXL2 in 6 patients, and it turned out it's pretty low, 10 to the minus 6, and so it looked like a good story. And we were able to do some quick functional studies to find out if there was RNA expression from 2 of the patients. We were surprised though. It seems like the RNA expression from 2 of the patients is essentially the same as for the positive controls, and so it doesn't look like these truncated variants actually are targeted for nonsense-mediated decay. So we're thinking that this could be a dominant negative mechanism. So for this one, we submitted this one change also for the model organisms to take a look and try and understand a little bit better what the mechanism of the pathophysiology of disease could be. And so, the UDN is a prime example of this precision medicine, and truly is, it goes beyond what we can do in the clinic and offers a lot of the tools that wouldn't be available to me even in the general clinic. And so the way this has turned out for us is whenever we're in the clinic as just the clinicians, doing an exome and we don't have an answer, we have a variant gene, we frequently actually will refer to the UDN, just knowing that there's just a lot more tools than we have available to us. And we've, part of the reason why Duke has a higher diagnostic rate for our participants is just, like I said, the gift of time, the gift of protected time and the resources and funding to able to do this work. And we're very interested in, development of targeted treatments, and of course, the model organisms work is going to be very interesting in that regard. Time and again, even though we have all the fancy tools like whole exome and whole genome sequencing, we come back to the phenotyping as being a key piece of information that allows us to really hone in on the genes of interest for these patients. And so we want to acknowledge the patients and their families, that none of this would be possible without them, and the team with Dr. Shashi and Dr. Goldstein as our PIs. And then our bioinformatician Nick Stong, who's based out of Colombia, who's been, quite instrumental in allowing us to evaluate some of these candidate genes. And we're happy to talk to you about any candidates that you might have, any patients, and you're welcome to contact Vandana, contact me, use the UDN number, or actually use the website. It's a web-based application that requires that the patient actually or the families do the application with a referral letter from the referring physician. So you can go online, you can Google Undiagnosed Network to get to the website. And I'll be happy to take any questions.
Do you guys accept patients into your study who don't have English as their first language?
We do,
Okay.
and we do accept patients, and there's resources that are available for people who don't speak English or do not have access to a computer or websites.
Okay,
Yes.
thanks.
Could you say a word or two about Gene Matcher? It's a web program that I'm not familiar with, but I have already applied for an account while you were talking. Sounds like an interesting, but how does it work?
I mean, essentially what you do is you submit you say, I have a variant in this gene. You don't even need to have the variant, and you put in your contact information, and then that's available. And so so people can just go to Gene Matcher and look up the gene of interest and find your name, your contact information, and they'll contact you. It's all over email. So then we start to compare notes and compare photographs. And it's one of those things that it's a low-tech approach, but has been incredibly fruitful for us.
So it's international,
Yes.
and you put in sequence data only?
No, you put in your gene name.
The gene name?
Just gene name. And then when people start talking to each other in private, you can exchange
So there's
variants.
no clinical data initially put in. You
Right.
just put in a gene name, and people say, oh, I'm interested in that gene, I'll talk to this guy.
Yes. It's too easy, right? Yes, and so the labs are actually entering data, so GeneDx will enter data, and it doesn't even need to be the clinician. So what happens is whenever we have a hit, GeneDx, the counselor at GeneDx, will contact the clinician and put us all in contact.